Label-free electrochemical detection in DNA biosensors. लेबल-फ्री इलेक्ट्रोकेमिकल डिटेक्शन में हम ऑक्सीडेशन और रिडक्शन के प्रोसेस पर रिलाय करते हैं इन डीएनए बायोसेंसर बिकॉज द receptor molecule is a DNA molecule and normally the target molecule is a DNA and RNA molecule. There is a process of hybridization. The process of hybridization is detected by the transducer and signals are generated. So we don't have to use any label molecule or any reporter molecule to detect the signal. Hybridization के दौरान रिडक्शन और ऑक्सीडेशन और रिडक्शन का प्रोसेस ही सिग्नल जनरेट करने के लिए काफ़ी होता है और देर इज़ नो नीड टू यूज़ एनी टाइप ऑफ लेबल मॉलिक्यूल. If you look at in this slide that this method is very simple, that a probe binds with its target molecule and during the process of binding, which is known as hybridization, there is a process of oxidation and reduction. Oxidation और reduction का ये process सिग्नल जनरेट करता है. हमें ये मालूम है कि ऑक्सीडेशन और रिडक्शन के प्रोसेस के दौरान इलेक्ट्रॉन्स की ट्रांसफ़र होती है. इस ट्रांसफ़र की वजह से सिग्नल जनरेट होते हैं जिनको ट्रांसड्यूसर फौरी तौर पर डिटेक्ट कर लेता है. सो द मेथड इज़ क्वाइट सिंपल. वी डोंट हैव टू यूज़ एनी लेबल्ड और reporter molecule. So this is a fairly simple method, but the signal generated are quite low. There are some drawbacks or disadvantages of this method. अगर हमने ये मेथड इस्तेमाल करना है तो हमें इन ड्रॉबैक्स को ओवरकम करना होगा. इनमें से पहला ड्रॉबैक जो है वी रिक्वायर्ड हायर पोटेंशियल. ये हम पिछले मॉड्यूल्स में पढ़ चुके हैं कि डीएनए ट्रांसपोर्ट द इलेक्ट्रिक करंट. And the second thing is that all the four bases of DNA are electroactive in nature. And guanine is one of the bases which could be easily oxidized. तो आम तौर पे DNA बायोसेंसर में गवानीन की ऑक्सीडेशन पर रिलाय किया जाता है. But the oxidation of this base required high potential. The second thing is there is a lot of high background and a number of other non-specific DNA oligos also binds with the target instead of target molecule. उसकी वजह दूसरे DNA samples में भी गवानीन के bases मौजूद होते हैं. इन bases की वजह से there is some non-specific binding. So how to overcome that? हमें ये तो मालूम है कि हम इन बायोसेंसर्स में यानी लेबल फ्री बायोसेंसर्स में कोई लेबलिंग के लिए मॉलिक्यूल इस्तेमाल नहीं कर सकते. देन व्हाट नेक्स्ट? दूसरी स्ट्रेटजी क्या है? द सेकंड स्ट्रेटजी इज वी कैन मॉडिफाई द इलेक्ट्रोड. सो देयर मस्ट बी अ मटेरियल, एन आइडियल मटेरियल which we could use for the DNA label free biosensors. The material which I am going to talk about is glassy carbon. We have discussed about this material in our previous module, but just for your revision, I'll discuss it. Glassy carbon is an allotropic form of carbon. It is highly pure. It has excellent stability in vacuum and in the air. At about 500 degrees centigrade, it is stable in the air. It is inert against different chemicals and it is inert against erosion. It is impermeable to gases and solution because this is non-porous, and it is significantly hard. Its hardness is comparable to the diamond, and it is a good electrical conductor. It is resistant to a number of to all organic and inorganic salts, and above all, it has a good biocompatibility. Now, we can use the glassy carbon as electrode, but still we need to modify this electrode depending upon the requirement of our DNA based biosensor. हम एक ऐसा बायोसेंसर डिज़ाइन करना चाहते हैं जिससे हम डीएनए की चारों बेसिस को डिटेक्ट कर सकें. तो इसके लिए हमें ग्लासी कार्बन को सेंसिटाइज्ड करना है. सो व्हाट टाइप ऑफ मटेरियल कुड बी यूज्ड टू सेंसिटाइज और इम्प्रूव द परफॉर्मेंस ऑफ द बायोसेंसर? इसके लिए हम डिफरेंट मेटल्स का इस्तेमाल कर सकते हैं. द फर्स्ट मेटल विच इज सिलेक्टेड इज कॉपर. Copper is an international conductor. It conducts electricity. It is a good selection. The other material which was selected by the scientists to develop this type of biosensor is cesium oxide. Cesium is a metal. It is an alkali metal. It has a large size. It has only one electron in its outermost shell. So this is a good selection. But there is another property of cesium which makes it very favorite. And that is that this element is one of those 5 elements which are near liquid or nearly liquid at the room temperature. So this compound basically adds softness to the biosensor electrodes. हमने इस इलेक्ट्रोड के लिए बायोसेंसर के लिए pH 7.4 सिलेक्ट की. हमें ये मालूम है कि pH 7.4 पर गुआनिन की ऑक्सीडेशन सबसे कम होती है बिकॉज दिस इज फिजियोलॉजिकल pH. So that is why we used copper and cesium oxide. We used 3% mixture of copper and cesium oxide in this electrode. This biosensor was successfully able to characterize and separate the all 4 bases of DNA. As you can see in this slide, we got different peaks for all those bases such as adenine, guanine, cytosine and thymine. For adenine and guanine, the peak range was between 0.8 to 1.2, while for cytosine and thymine, we were able to detect them around 1.3 and 1.5. इस कहने का मतलब ये कि if we make some sensible changes or thoughtful changes in the designing of a biosensor, we are able to detect our target molecule. Now we will discuss another important example. In this example, the scientists used graphene as an electrode, and in this case, the design was to detect the HIV target DNA. The single-stranded DNA was used and it was immobilized on graphene by using pyrene. Pyrene is an organic compound. It has 4 benzene rings. So benzene ring, इसमें 4 benzene rings हैं. Pyrene के साथ DNA के probe को attach किया गया and then this was attached on the graphene surface, and this 2D material of graphene was very successful in detecting the HIV DNA. You can see in this slide that the electrochemical signals detected by the binding of PNA, इस biosensor में हमने PNA का इस्तेमाल किया. PNA is peptide nucleic acid, जिसके हम मुतालिक हम पीछे डिस्कस कर चुके हैं, and this PNA was able to detect the HIV target DNA. So the biosensors which are label-free, based on label-free electrochemical detection in DNA, are very successful. They are simple, rapid, easy to use, they are highly sensitive, but still we need some sort of modification to use these biosensors as per requirement, and they are very stable. So in summary, the label-free electrochemical detection is one of the suitable methods. In this method, the main advantage is that we don't have to use any labeled molecule. The only thing which we have to do is the modifications of the electrodes, because the electrode can can amplify the electrochemical signals, and these signals can be read as separately read. The modified electrodes are used to amplify the signals and reduce the noise. The glassy carbon electrode and graphene are the 2 ideal types of electrode materials which are used in the preparation of label-free electrochemical detection in DNA biosensors.